Light is a key feature of our shared, objective world, and exists in more forms than there are ways to prepare a potato. However, as visually sensitive as we are, our human eyes can only detect a limited range of light. Thankfully, technological advancements allow scientists to detect and measure several other forms of light that are invisible to the naked human eye, such as ultraviolet, infrared, and radiation waves. Over the last century, yet another unique form of light has been identified called ultra-weak photon emission, UPE. And recently 2 groundbreaking studies have discovered evidence that all living organisms emit UPE, that our brain emits the brightest light, and that this light disappears upon death. Experts have some ideas about what creates UPE, but no one knows for sure. Beyond the sun or other cosmic sources, life itself can also emit light. You're likely already familiar with the high-intensity, sometimes eerie illumination of bioluminescence, which emerges as a chemical reaction within a living organism. Many life forms, including bacteria, fireflies, and countless creatures inhabiting the deep dark oceans, emit bioluminescence. Some might even say that bioluminescence is the most common language on Earth. Meanwhile, everything in the known universe that's warmer than absolute 0 degrees, including you and me, emits thermal radiation, a type of infrared radiation, or more commonly known as infrared light, which is invisible to us. However, the light known as ultra-weak photon emissions, UPEs, is a distinct phenomenon from thermal radiation or bioluminescence. As its name suggests, UPEs are a super low-intensity light that is so faint, it exists 3 to 6 orders of magnitude lower on the spectral range than the human eye can see. The common consensus is that UPEs are a continuous stream of photons that emerge as a byproduct of biomolecular reactions in living cells that generate energy, which is why UPEs are sometimes referred to as biophoton emissions, or simply biophotons. In other words, as living cells generate energy through metabolism, they produce oxygen molecules as a byproduct that have excited electrons. When these excited electrons return to a lower energy state, they release photons, light, through a process called radiative decay. Biophotons were first discovered in 1923 by a Russian and later Soviet biologist and medical scientist named Alexander Gurvich, after he conducted various mitogenetic experiments, such as placing photon-blocking barriers between onion roots, which prevented the plant from growing. During the 1970s and 1980s, German physicist Fritz Albert Popp coined the term biophotons after completing a series of thought-provoking yet controversial studies that suggested living cells continuously emit photons. After completing over 150 studies and scientific journal papers, and writing 8 books that address various questions related to biology, complementary medicine, theoretical physics, and biophones, Popp believed that UPEs could be involved in communication between cells, coherence, and possibly even help regulate biological processes. While Popp's hypothesis remains debatable, in the decades since, the faint light of UPEs has been found in a wide range of life forms, from tiny single-celled organisms and bacteria to larger plants and animals, including humans. In fact, a growing body of evidence suggests that humans are quite luminescent, from as early as the moment of conception. Now, thanks to the 2 new studies I'll tell you about soon, we know UPEs glow the brightest in the brain, that they change based on various cognitive tasks, and they only stop glowing after we die. The glowing brain. The idea that UPE biophotons emerge as a byproduct of radiative decay suggests that the more energy a tissue burns through metabolism, the more light it should give off. Considering this, and inspired by Gurvich's work, biophysicist Neuroscience Murugan at Wilfrid Laurier University in Ontario rationalized that our brain, which is an especially metabolically expensive organ, should emit the brightest UPE glow. Furthermore, previous research on biological tissues, such as neurons in a petri dish, has detected a faint but continuous glow of UPEs involving as few as a few photons to several hundred per square centimeter per second. So, as Murugan told Connor Feely of Scientific American, scaling this up to humans, we wanted to know if those photons might be involved in some information processing or propagation in the brain. To find out, Murugan led a collaborative team of scientists to investigate and then published their small but groundbreaking study on March 21, 2025, in the journal iScience. Since the brain is encased within our skull, and UPE has only been found in living organisms, Murugan and her team needed to think creatively to determine whether they could measure the UPEs of a living human brain from outside the skull. Experiment 1. To achieve this daunting task, the researchers recruited 20 participants, 10 women, 9 men, and 1 non-binary individual, between 19 and 52 years old. Then, they had the participants sit in a blacked-out room while wearing electroencephalography (EEG) caps, which uses electrodes to measure the brain's electrical activity. In an attempt to detect UPEs emitted from the participants' brains, the team also positioned photon-amplifying tubes around the participants' heads, clustering them over the regions responsible for our visual, the occipital lobes in the back of the brain, and auditory processing, the temporal lobes on each side. Additionally, the researchers set up separate UPE detectors facing away from the participants to distinguish the UPEs emitted from the participants' brains from other photons in the room. During the experiment, the researchers recorded the brain activity and UPEs of participants at rest, and as they performed sound-based tasks, since they were sitting in the dark. The results. The study's results were as incredible as they were conclusive. As Murugan told Feely, the very first finding is that photons are coming out of the head. Full stop. It's independent, it's not spurious, it's not random. As for distinguishing the UPEs coming from the participants' brains from background photons in the room, the researchers write in their study, "We view the current results as a proof-of-concept demonstration that patterns of human brain-derived UPE signals can be discriminated from background light signals in darkened settings despite very low relative signal intensity." These results make the scientists the first ones to discover that the brain's UPEs are strong enough to be detected from outside the skull. Experiment 2. After the results of the first experiment, Murugan and her team wanted to see whether the intensity of the brain's biophotons changes when people perform various cognitive tasks. So, the team then repeated the experiment, but incorporated additional tasks for the participants to complete, such as opening and closing their eyes. Similar to before, Murugan reasoned that since the brain demands such high amounts of energy, the UPE intensity should theoretically grow when people are engaged in high-energy tasks, such as visual processing. Such an idea seems even more reasonable considering that's basically what's already been observed among neurons in a petri dish. The more active the neurons are, the more UPEs they emit. The results. This time, however, the team's hypothesis didn't pan out. They didn't detect any increase in biophotons during moments of higher EEG activity levels. This suggests there's a significant change that occurs at some point between a few cells in a petri dish and a whole living brain. Murugan suggested to Feely that maybe UPEs are not getting picked up by our detector because they could be getting used or absorbed or scattered within the brain. That said, the researchers did find another more exciting surprise. While UPE activity didn't increase whenever the brain's electrical activity did, the UPEs did change in response to one thing: whenever the participants transitioned from one cognitive task to another. In other words, instead of increasing during each task, the UPEs increase while switching between tasks. This result suggests a connection between the brain's processing and the biophotons it emits, and it piques the researchers' curiosity about the role UPEs play in the brain. In the future, Murugan and her team's research has sent shockwaves through the scientific community and is already inspiring a range of ideas for future research into neural UPEs. For instance, Daniel Remondini, a biophysicist at the University of Bologna in Italy, told Feely that the new study makes him wonder, how far can these photons travel inside biological matter? Yet another curiosity is whether each person's UPEs are as unique as a fingerprint. If so, future research should include baseline UPE measurements from participants to accurately interpret potential anomalous activity. On April 24, 2025, a month after Murugan and her team's study was published, a different but equally astounding study was published in the Journal of Physical Chemistry Letters by physicist Vahid Salari from the University of Calgary, Canada, and his colleagues. The researchers were curious about exploring the biological significance of UPEs, so they focused their study on analyzing and comparing UPEs emitted by mice during life and death. They also monitored the effects of chemical treatments, injury, and temperature changes on UPE in two types of plants, Thalia cress, Arabidopsis thaliana, and dwarf umbrella tree, Heptapleurum arboricola, leaves. The scientists designed ultra-dark enclosures for the mice and plants during their experiments to eliminate potential environmental light interference. They then used specialized cameras to monitor the UPEs. They used an electron multiplying charge-coupled device, EMCCD, capable of detecting and quantifying single-photon events without the need for an image intensifier to record UPEs from thalocress and dwarf umbrella tree leaves. In addition to imaging healthy leaves, the team also caused physical injuries to some, and exposed others to chemical agents to see if UPEs changed depending on an organism's health. For the mice, the team used a charge-coupled device, CCD camera with an IVIS system, which captures images by converting photons into electrons. This tech allowed the researchers to compare even the faintest biophoton emissions between living and deceased mice. The results. They found that injuring the plant leaves, either by causing physical injuries or exposing them to chemical agents, revealed greater light emissions in the damaged part of the plants compared to healthier areas. As you can see in the image below, the brighter white and yellow areas are where the leaves were damaged. The researchers believe their findings provide strong evidence that reactive oxygen species could indeed be responsible for the soft glow. The team found similarly astonishing results when imaging the mice. There was a distinct difference in UPEs between living and deceased mice, with living mice emitting robust emissions while the UPE glow in euthanized mice dropped until it was nearly extinguished. These combined results demonstrate that monitoring UPE has the potential to help scientists measure and assess the vitality of animals and the stress response in plants, and that the death of an organism results in the cessation of biophoton emissions. In other words, our light really does go out when we die. Perspective shift. Thanks to technological advancements, scientists can now investigate some of the more mysterious aspects of our living existence, such as consciousness and life itself, and reveal new layers of complexity and intricacy, while also confirming phenomena we instinctively, though perhaps subconsciously, refer to. I mean, we've long used words like glowing or shining to describe a particularly happy or healthy person, and as I just mentioned, when people die, we talk about their light going out. Come to think of it, when people are unwell, we may say their sparkle is gone. Now it appears that there may be more truth to these metaphors than we realize. Meanwhile, Murugan and her team's research adds new insights into neuroscience by being the first to document UPEs glowing through the human skull. I wonder how much brighter the UPEs are in our heads compared to the rest of our bodies. I'm also curious whether the brain's high metabolic energy demand is behind the increased glow, or whether there's another reason. For instance, might UPEs have any connection to consciousness? Some neuroscientists believe consciousness uses quantum entanglement. If so, might UPEs also be involved? And what might that mean, considering UPEs are found in all living organisms? Could that add additional evidence toward theories like panpsychism? Additionally, the combination of both studies raises my curiosity about whether there are any connections between the fading of UPEs at death and the so-called wave of death that neuroscientists previously observed when a person dies. All that said, I'm glad scientists don't think UPEs are simply a byproduct without any function of their own. Such an assumption would conflict with what we know about nature, which is that nature is the ultimate recycler, wasting nothing. Now, thanks to technological advancements, scientists can now investigate some of the more mysterious aspects of our living existence, such as consciousness and life itself, and reveal new layers of complexity and intricacy, while also confirming phenomena we instinctively, though perhaps subconsciously, refer to.